DNA is two strands, both run in a 5 to 3 feet direction that are anti-parallel. Each phosphate group links the 3' carbon of one sugar to the 5' carbon of the next sugar along the backbone. T and A pairs have 2 hydrogen bonds. C and G pairs have 3 hydrogen bonds. Pairs of complementary bases form hydrogen bonds that hold the 2 strands of the DNA double helix together. That's what creates a double helix DNA structure. This DNA model was ripped apart to create a new DNA strand and put back together by new floating base pairs that will put the old DNA strand back together while it's creating the other new DNA strand. The DNA helicase is an enzyme that unwinds the double helix by breaking the hydrogen bonds between the complementary bases to create a single-stranded RNA structure. The RNA polymerase unwinds the double helix DNA strand to create an RNA strand by adding ribonucleotides to the template strand. When it's done, it cuts off the new RNA strand, and it zips itself back up, and it continues its cycle. After the DNA is done making the mRNA strand, the mRNA, which is messenger RNA, it leaves the nucleus through the tiny pores, and a ribosome comes and it takes every 3 base pairs and it makes them into protein, which we use for our body because the DNA strands cannot fit through the pores in the nucleus, so it has to be protected, and that's what translation is. During DNA, a ribosome comes and it matches up the 3 letters for RNA that goes under the DNA strand, and it takes the 3 letters that make up an amino acid, 3 groups, and it makes a different word that makes an amino acid, and it keeps going and going and going.